So last class we spoke about the callus and we told that callus is obtained from an explant. An explant, I told you, it can be any part or piece of the plant. So when we provide the exact nutrition, particularly a growth hormone and environmental conditions, we can form calluses. And today we shall go a step ahead and see what is the anatomy of the calluses. Last week we saw that how we can initiate, last class we saw that how can we initiate a callus. And today we shall look into the anatomy of the callus. So we define callus as an undifferentiated, unorganized mass of parenchymatous tissue. They are amorphous in the form. We told that the texture of the callus and the pigmentation of the callus, look at this, the color of the callus, that is the pigmentation of the callus or the texture of the callus, all depends on your explant. What type of explant you have chosen, what is the species of explant you have chosen. Based on that, we get this type of different colors and the textures. And also remember, we told that callus is just not formed in the laboratory. It can be formed by the nature also when there is an infection from the microorganism or by some insect or some mechanical and physical harm caused to the plant. That time also callusing can occur. So we shall look into the anatomy of the callus. So we define a callus as a homogeneous. See that what we mean to tell is, like we have told that it is an undifferentiated, unorganized mass of parenchymatous cells. But when you look at a callus, it is not homogeneous. It is not same. Okay. So a homogeneous callus consisting entirely of parenchymatic cell is rarely found. So we can't find the entire callus truly having a parenchymatous cells. There is some sort of differentiation. Okay, like I told you, tracheal cells are present in the callus. So that is the meaning of your first sentence. They cannot be truly parenchymatous. Now let us define a word cytodifferentiation, which is very important for callus. So what do you mean by cytodifferentiation? Cyto means, all of you know that it is a cell and the cell has to differentiate. It has to undergo morphogenesis into different types. It has to undergo the cell division and finally form a whole plant. This is called as cytodifferentiation. The morphological development of undifferentiated cell into more specialized one is called as the cytodifferentiation. So undifferentiated, whose here are undifferentiated? It is our callus. Okay, we told it is an undifferentiated mass of tissue. But what happens? This undifferentiated mass of tissue should undergo certain changes so that it can grow into a specialized cells. So that is what is our goal or aim is. Once we develop a callus in the laboratory, our duty is just not to maintain the callus as itself. Okay, sometimes you require the callus in its natural form. So there are other means. But my main idea is like from the callus, if I want to develop a whole plant. And so what should I do? I should see that my callus undergo differentiation. That word becomes important. So from undifferentiated mass of cell, it should undergo differentiation, it should undergo de-differentiation, and it should form a specialized cells or organs. So what happens is, like we told that it is having some amount of vascular elements. These vascular elements formed by the meristematic cells. It can be formed from your meristematic cells of your root tip or stem apex and vascular cambium. Look at this word meristemoids. Okay, so these callus, they'll be having a small nest of dividing cells. Okay, if this is my callus, they'll be having a nest of dividing cells somewhere here. Okay, and they form a vascular nodules. Okay, and these are called as meristemoids. So my callus is having a vascular nodules, which we term it as a meristemoid. What is a meristemoid? A small triangular stomatal precursor cell that function temporarily as your stem cell. Okay, so what happens, like from the stem cell, different organs are being formed. Okay, your hands, legs, or whatever your anatomy of your body is formed. Similarly, in the plant also, in the callus, what we call it as a meristemoids, which are nothing but this, it represents the stem cells in the meristem, and this is a precursor. Okay, precursor means which is going to initiate into the formation of the shoot or root or embryo or any other part of the plant. So this becomes very important. So if there is a meristemoid, it is going to behave as a stem cells, and these stem cells are the precursor cells. Okay, their function is only temporary. And what is their function? It is to produce shoot apexes, root primordial, vascular cambium. These are the functions of it. So what is happening? From an undifferentiated mass of cell, it is going to develop into all this because of this meristemoid or because of this precursor cell. So always remember, whenever you come across this word primordia, okay, it is the earliest stage of development. The starting stage of the development is known as the primordia. So this is called as a primordia. From this, other parts can develop. So how are you going to subculture a callus? You cannot keep a callus in the same media and you cannot grow it. You have to subculture it. So another important question, what do you mean by subculturing? So once the callus tissue is formed from the explant, okay, the callus is transferred to the fresh media, and this is known as subculturing. So I have an explant. From this explant, what did I develop? I developed a callus. Okay, I developed a callus. Now, what you have to do? This callus, okay, it is transferred into a fresh media. Here it is. It is transferred into a fresh media. So how long you should transfer it to a fresh media? It takes approximately 3 to 6 weeks. So remember, after the callus has been grown from the explant, it becomes necessary to subculture the callus into the fresh media, while still it is in association with the original tissue. Who is your original tissue here? My original tissue here is the explant. So this is the meaning of subculturing. So you grow that explant, develop it into a callus, and you're growing that in the nutrient media containing the growth regulators. After a period of 3 weeks or 6 weeks, what you're doing is you're transferring this callus into your fresh media. And this fresh media, no, it will have all constituents same as the previous media, only that it is newly prepared, freshly prepared. Why are we doing this? Let us see that. So when you're growing your callus in the same media, that is your first media in which you have put your callus, what happens is there is a depletion of the essential nutrients. So when the callus is growing in the media, what happens? It uses up all the nutrients in your media. So what happens? The nutrients comes down. There is decrease in the nutrients. Why? Because the callus has used up the nutrients. And also what happens? We can grow callus in the solidifying agents. What are your solidifying agents? They are your gelling agents. What is my gelling agent? It can be agar. It can be your agarose. It can be your gelatin. Particularly, we are going to use the agar. So what is a gelling agent? Means it is a solidifying agent. It just solidifies your media and nothing else function it has. It is just an inert material. Okay. So what happens? Desiccation of your gelling agent happen. Desiccation means drying. Okay. See, if this is your conical flask. Okay. If this is your conical flask and this is my media and here is my callus, what happens? It starts desiccating. It becomes dry and it will be only this much. No more to the walls your media is present. Okay. So what happens? That is why we go for the fresh media. So growth on the same medium for an extended period will cause depletion of essential nutrients and gradual desiccation of your gelling agent. Therefore, it is necessary for me to transfer my callus along with its associated original tissue into the fresh media. Apart from these two problems, I have another problem. What is that? The metabolites secreted by the growing callus may accumulate to toxic level in the media. So we told that naturally every plant will produce a metabolite. This is a separate chapter. You'll be studying in detail the metabolites. Okay. So I told you, I took an example of apple and I told you like it turns brown in color. Why it turns brown in color? Because there is polyphenolic acids which are present there. Phenolic compounds are present there. Flavonoids may be present there. Terpenes can be present. So these are the metabolites which are secreted by your plant. But what happens if they are over secreted in that same media? It leads to a toxic level. Profusely what is happening? The metabolites are being produced by the plant and you are not removing that metabolites. So what happens? These metabolites will accumulate into your media and it reaches a level such a way that it becomes toxic to your own callus. That is why I go for a fresh media. Now, when I'm telling that I will take my callus, put it in the fresh media so that it grows again well. So you cannot do it as your wish. There is a particular protocol or procedure for transferring. Okay, of course, you do all aseptic methods. That is right. But what is another important point you have to remember is the transferred fragment of callus must be of sufficient size. It should have a definite size so that it can grow in the fresh media. I cannot take a large callus and put it into the media or take a small piece of callus and put it into the media. Both can have a negative effect. Okay, so the sufficient size becomes important. So you can see here, if I'm transferring a small inoculum of my callus into the media, into the fresh media, my callus may not grow. Otherwise, it will grow very slowly. So which is not advisable. Therefore, a scientist named as Street recommended that the inoculum here means your callus, be at least 5 to 10 mm in diameter and weigh 20 to 100 milligrams. So your callus should be at least 5 to 10 millimeters in diameter and its weight should be somewhere around 20 to 100 milligram, so that you can transfer that callus from this, okay, to your fresh media. So this is what is known as subculturing of the callus. So once the callus tissue is formed from your explant, the callus is transferred to your fresh media along with your original tissue. This process is known as subculturing. Why should I go for subculturing means? Because my media, there will be depletion of the essential nutrients because my callus is taking up all the nutrients. Otherwise, there can be desiccation or drying of the gelling agent or my agar. Otherwise, the metabolites produced by my callus will accumulate in the media and it will reach to a toxic level that my callus will not be able to grow. That is why I transfer my callus into the fresh media. When I am transferring my callus to the fresh media, I should take certain precaution. I should follow all the aseptic condition, sterilized condition, and the most important is the size of my callus. The size of my callus should be 10 millimeter in diameter and the weight should be somewhere around 20 to 100 milligram. Otherwise, my callus will not be able to grow in the fresh media. So when are you going to perform your subculturing? That is your next question. I have grown my callus. Now when should I do the subculturing? Grow it in a fresh media. Successive subcultures are usually performed 4 to 6 weeks with the culture maintained in an agar media at 25 degrees centigrade or above. So you do your subculturing for every 4 to 6 weeks and see to that you maintain your callus in the agar media and you provide your temperature at least 25 degrees centigrades or above, or usually 25 degrees centigrade plus or minus 2. And you see to that, depending on the species, what type of photo period I should give. Should I give 16 hours or should I keep it in the light or should I keep it in completely dark? That all depends on the species, what you choose. Now comes this word, friable callus. Okay, so we are more interested, the scientists are particularly more interested in a callus which is soft, which is friable. What is it? We shall look into it. A friable callus can be subdivided with a thin spatula or scalpel. So whenever I want to do further experiment with my callus, forget about developing a new explant. Suppose say I want to produce a single cell. You studied that experiment, okay, wherein we kept a callus, above that we kept a filter paper and we kept a small cell and we saw that the small cell was able to grow into a new callus. Okay, so whenever we want to grow a single cell, we usually use a friable callus. Why we use a friable callus means because this callus can fall apart easily and can be used to generate suspension culture. So you can disassociate easily. For example, this is my callus. I can just cut this callus very easily by using a thin spatula or a scalpel. So it can easily disassociate. Okay, so I can get small pieces of it. I can get a single cell out of this friable callus. It's easy for me to disassociate it. So it can fall apart easily. So how I'm going to cut my callus? By either using the spatula or I can use the scalpel, put it in a sterile petri dish, and then you can use your scalpel to cut it into small pieces. Remember, whenever you are using callus for any type of future experiment, you should use only healthy tissues. And brown color or necrotic tissue must be discarded. The problem of tissue culture, you studied the brown color. We saw that how the explant was developing the brown color, the phenolic compound, your metabolites, it was releasing into the media and ultimately killing the plant itself. So whenever it is brown in color, please discard it. Whenever it is a necrotic tissue, remember that the dye tip example we spoke about in problems in tissue culture. Necrotic tissue means that is, they have lost their ability to grow. They are dying. Okay, such type of necrotic tissue also must be discarded. These are very important precautions we should take it in the both plant and animal tissue culture. When you don't find that the tissue is healthy, when you find that they are contaminated, when you find they are weak, please immediately discard it, because you cannot proceed with the experiment, because these are very lengthy experiments, and finally you cannot tell that nothing has grown. Okay, so these precautions must and should be taken care of. So now there are many methods, okay, wherein you can develop an alternate methods for long-term maintenance of the culture. So if I want to maintain it for a year, more than a year, what can I do? I can freeze them. Okay, there are techniques like cryopreservation, freeze preservation. So I can store them for a long period of time. So in this slide, what we spoke was, we told that we can go for subculturing for every 4 to 6 weeks, and we should maintain a proper temperature of 25 degree centigrade plus or minus 2. And particularly if I want to carry out suspension culture, next shall speak, that is a separate chapter, suspension culture. That is, I'm growing single culture, deaths or single cell. Then what is important is, it is important my callus should be friable. That is, it should easily fall apart or easily should be disassociated. And I always should choose a healthy plant for doing any type of transfer experiment. If I want to long time storage of my explant or whatever tissue culture plants or even callus, I can always go for freeze preservation, depending on what type of explant you're using. See, this is a very good example of your callus, how they are growing it. Look at this. This is your explant. Okay, I'll simply explain to you all this part. It is quite interesting. Freshly cut pieces from surface sterilized plant or any organ, okay, they are placed in the nutrient media. This is your nutrient media. It has agar. Okay, so here it is the leaf plant, the leaf which we have used. So it is placed on the nutrient media consisting of suitable proportion of auxin cytokinins to initiate culture, so that it can develop into a new plant. So the explants on such media exhibit calyxing. See, I have cut the plant under all aseptic condition. I have placed it into my solidified media. I have added my cytokinin and auxin, and then you see my explant turning into a callus. Okay. So you can see here from where the callus is being developing. Clearly observe this. This explant on such a media exhibit callusing at the cut ends, which gradually extends to the entire surface of the tissue. The callus is separated from the explant and transferred to the fresh media of the same composition to enable to build up the mass of tissue. So repeated subculture on agar media improves the friability of the callus. That is what we were speaking. This is a small picture which shows that. Okay, so I have cut the explant. For example, here it is a leaf. I have provided the nutrients. I'm growing it in the agar media. I have provided the growth regulators, auxin and cytokinin. And you can see at the cut edges of my explant, it is started to callus and the entire callus can be formed. And then I can subculture it in the same constituent of the media. Whatever the first constituent I have used, freshly prepared media also should have the same constituents. Okay. When I'm doing such type of subculturing, I will get the friable cultures, which can be easily disassociated. So this is an example of suspension culture. We'll not go into it for now. and this is simply showing an plantlet. Okay, like how small plantlet can be used as an explant for growing into the callus. So let us look into the why are we doing this callus culture? What is its importance? We shall look into that. So the first one is it helps in the production of secondary metabolites. So I told you the secondary metabolites, you have primary and secondary metabolites. Okay. Do not worry about it now. But just remember that your plants produce secondary metabolites which are economically useful for mankind. Okay, I told you the medicinal plants, the flavonoids, the terpenes, the saponins, the alkaloids, phenols, flavonoids, whatever name them, is all your secondary metabolites. Okay, so these are very important. They are secreted by the plant and it is important for us. So what we do is we will try to extract all the secondary metabolites. So it helps us in the production of the secondary metabolites. When my plant is producing secondary metabolites, what will I do is I will extract all the secondary metabolites for my use, for the use of mankind. For that we use the callus. Second is it is useful for synthesizing starting compounds that are subsequently modified to yield a desired product. That is, I have taken the explant, I have produced a callus. Okay, from the callus, then what I can do, whatever the decided, if I want, I can develop a shoot, I can develop only the root, or I can develop a whole plant. So by the callus, I can modify it to yield a desired product. I can genetically modify my callus. I can induce a gene into it, a new gene which was not present. So I can get a desired product out of it. So that is the meaning of your second application. Starting compounds that can be subsequently modified to yield a desired product. It is the starting material for your vegetative propagation. That is, if the plant is not undergoing any sexual, we do not want the sexual propagation, we can always produce a vegetative propagation by using the callus culture. One of the methods for producing vegetative propagation. Callus is the starting material of your suspension culture. So what is the suspension culture means? Just remember for time being, it is to obtain a single cell. Remember the nurse cell experiment, okay? To obtain a single cell, we use this callus. Okay, you have obtained the single cells. So what are you going to do with it? I can do many things about it. When we come to the chapter, we will study. But another method, by using suspension culture, I can study the morphology. I can study the biochemical changes. I can study the metabolic pathway. How the cellular metabolites is occurring in my plant. I can study all this by using suspension culture. So by using callus, I can start off with the suspension culture. Suspension culture consists of the friable callus, that is single cell callus. By using the single cell callus, I can study the morphology of my plant. I can study the physiology of my plant. I can see what are the biochemical reaction it is happening, how a root is growing, how a shoot is developing, what are the cellular metabolites are present in that plant. This all I can study by using the callus culture. One more point, see girls, the students, that whenever we are multiplying a callus, okay, like when we are growing it indefinitely, I told you that we should follow subculturing. But sometimes, most of the time, what it can happen is your callus loses the morphogenesis. Particularly if your callus is too old. Okay, I told you this is very important, this part. It should be a healthy tissue. So it can be multiplied indefinitely. Your callus can keep on be multiplying. You can maintain it in the callus form throughout when you're doing subculturing. But most of the time what happens is it can lose its morphogenesis, particularly if your callus is too old, or it depends on the type of species you are forming the callus. Okay, so callus varies from species to species, that is from explant to explant, from which explant you have taken. If I've taken from A explant, I cannot expect even my B plant should produce the same type of callus and it is also healthy. No, it cannot happen. So species to species also it varies. So finally, please remember this terminology for the examination. What do you mean by organogenesis? See, a callus tissue is a good source from which the whole plant can be regenerated by changing the nutrient and the hormonal constituents in the culture media. This kind of plant regeneration is known as organogenesis, or it is also known as embryogenesis, where the cells will form an entirely new plant. So a callus tissue, from this callus tissue, I am regenerating a whole plant. How? By providing the nutrient, by changing the hormonal constituents. And this process, wherein your undifferentiated, unorganized mass of your callus, when you're supplying it with proper growth hormones, it can develop into an organ, is known as the organogenesis, or it is known as the embryogenesis. So organogenesis means the development of the adventitious organ or primordial. Primordial, I'm told you, is a developmental stage. A small triangular shaped cell which can develop into a shoot, root, or whatever, okay, from undifferentiated cell mass. Okay, this undifferentiated means from the callus to a tissue by a process of differentiation. So an undifferentiated mass of tissue which is able to differentiate and de-differentiate and develop into an organ is known as organogenesis. Another terminology is calliogenesis. Calliogenesis is the type of organogenesis by which only adventitious shoot bud initiation takes place. Only you're developing the shoot. From where? From the callus. How? By adding your nutrient, cytokinin. What is rhizogenesis? Is a type of organogenesis by which only adventitious root formation takes place in the callus. So, you know what is organogenesis? You know what is calliogenesis? That is shoot bud formation. When I form the root formation means it is rhizogenesis. What is meristemoids means? Meristemoids, I told it is a triangular shaped nodules which are present. Okay, these meristemoid is a localized group of meristematic cells or the stem cells that arise in the callus tissue and may give rise to the shoots and the roots.